Hello and welcome back. In this video we'll be discussing Chapter 4 of Garrett Biochemistry, which deals with amino acids. So in this chapter we're going to discuss the structure and properties of amino acids, and we'll get to the overview of this chapter in just a minute. So amino acids exist as enantiomers, and we discussed what an enantiomer is in organic chemistry. So you should have learned about what an enantiomer is, and that is a mirror image of a molecule that has a kind of handedness, or it is not superimposable on its mirror image. So amino acids do exist in mirror image forms, and only the L isomers of amino acids are commonly found in nature. There is an amino acid that isn't chiral, and that's glycine, and we'll discuss that as we get to it. So why are amino acids uniquely suited to their role as building blocks of proteins? That's because of their structure. So amino acids have an amine group, and this is generally drawn on the left. This is the notation for drawing an amino acid, and a carboxylate group that is, or carboxylic acid group that is drawn on the right. And then in the center we have what's called an alpha carbon, and that is a chiral carbon or a tetrahedral carbon that exists, that can exist as different conformations, obviously. And so the R group in the L amino acids, and we'll talk about what that means in a little bit, is going to be pointing out at us, and where there's going to be a hydrogen pointing back if you orient your amino acid in this way. And this is the way they are generally drawn in proteins, going from the amine end to the carboxylate end, as we discussed in an earlier chapter, that there is a direction for drawing these molecules. So the outline for this chapter, the things that we're going to discuss are: what are the structure and properties of amino acids? What are the acid-base properties of amino acids? What reactions do amino acids undergo? What are the optical and stereochemical properties of amino acids? What are the spectroscopic properties of amino acids? How are amino acid mixtures separated and analyzed? What is the fundamental structural pattern in proteins? So we'll begin to answer these questions in this chapter, but amino acids are so integral to biochemistry that we'll continue answering these questions as we move throughout the course. So again, amino acids contain a central tetrahedral carbon atom that's called the alpha carbon. There are 20, or some charts give you 21, common amino acids. Amino acids join via peptide bonds, and we will discuss what that means in this chapter. Rarely, several other amino acids, not the common 20 amino acids, can occur in proteins, and some amino acids are not even found in proteins. So what are the structures and properties of amino acids? We have the, as I drew earlier, amine group on the left, the carboxyl group on the right. That's the notation for drawing these. If we orient that molecule this way, the side chain, or the R group, will be pointing out at us, and the hydrogen will be going back. Now that R group, and that's for an L amino acid. Now that R group can vary, and we'll talk about what the differences are. So that's the thing that varies in amino acids. The amine group and the carboxyl group are the functional groups that join in peptide bonds. So the amine group will join to a neighboring carboxylate group, and the carboxylate group will join to a neighboring amine group, and that is why these are good at forming polymers. So here's a ball and stick model, which is another way of representing this kind of molecule. And you'll notice that amino acids are tetrahedral. So the alpha carbon in the center is a tetrahedral carbon having a tetrahedral 3D shape. So this is the anatomy of amino acids except for proline and its derivatives. And glycine is also different in that it only has it has a hydrogen as an R group, making it achiral, and we will discuss that in just a little bit. Okay, so two amino acids can join together and they react with the loss of a water molecule, which is a dehydration reaction, and when they do, they form a covalent bond. The bond joining the two amino acids is called a peptide bond. So you can see here we have the amine group. This is a nitrogen. Let me change my color here a little bit. Let's make it yellow. Okay, so this is a nitrogen, right? This is a carboxylate group, so we got a carbon double bonded to an oxygen and another oxygen. Okay, and here in the center we've got our alpha carbon. We have our R group sticking out. Again, that's going to vary. And so we've got that next to its neighboring amino acid. So this is just a different amino acid where we have our R group, amine group, our carboxylate group. Okay, all of those are going to be the same, but the R group varies, and those join together with the loss of water. Okay, so what are we losing? We're losing an oxygen and two hydrogens, and we're going to be joining the carbonyl group to the nitrogen, which retains one hydrogen. This is called, when that forms, that's called a peptide bond when it forms with amino acids. Okay, with amino acids it's called a peptide bond, and that bond has the functional group of being an amide. So that NHC double bond O, that is an amide bond. You may have seen that in organic chemistry. And I'll just mention this real quickly. Amide bonds are quite stable. They're not very reactive, and we'll look at why that is in this chapter. But that's because they're essentially sp2 hybridized bonds. They have resonance, and that makes that amine group, which was formerly the amine group of the amide, not basic or acidic. Okay, and so they've lost their basicity. The amine group has lost its basicity. The carboxylate group has lost its acidity, and it has become essentially unreactive, giving us a very stable backbone for a polymer in a protein. Okay, so in this chapter you definitely need to learn the names, the structures, the pKa values, and the 3-letter and 1-letter codes for the amino acids, and we'll talk about what those are. Okay, so we're going to talk about in this particular video the different types, the different categories of amino acids. We have non-polar amino acids, polar uncharged amino acids, basic amino acids, and acidic amino acids. Okay, so let's look at this picture here that I've brought up, and we're going to see this picture again in a few minutes. I just grabbed this off of Google. And so what we see here are the 20 or 21 amino acids that are naturally occurring, and here they are. Here we have electrically charged side chains at the top in this light gray or light blue. And so notice here, okay, here is our we can't draw. Oh yeah, we can. Here is our alpha carbon, okay? Here's our amine group and our carboxylate group. Okay, so where is our R group? Our R group is right here, okay? Now, these amino acids on the left have a nitrogen that can pick up a hydrogen. They were basic, and when they pick up a hydrogen, they become positively charged. So don't get confused about that. They were bases that under biological conditions usually pick up a hydrogen to become positively charged. Okay, so here on the next one we have our amine group, we have our alpha carbon, we have our carboxylate group, and here is our R group. Okay, so for this one we have histidine. So the first one was arginine. Okay, now its three-letter abbreviation is, I don't know if I can make this smaller. Let's see. Yeah, let's make it a little smaller. Its three-letter abbreviation is, oh boy. Oh, can't write over there. ARG. So you see that right here. Okay, and its one-letter abbreviation is R. Now, some of these don't have the abbreviation of the first letter because we have 20 amino acids and some of them have the same first letter. So here we have arginine, down here we have alanine. So R is used to distinguish it from the other amino acids. And so if we form a chain of these amino acids to make a protein, we're going to see chains with abbreviations of one-letter codes or three-letter codes. For example, let's say we have arginine, histidine, lysine, okay, then it's that we would see this three-letter abbreviation if these guys are connected together, or we can have RHK. Notice K is for lysine, and that's why you really need to learn these three-letter and one-letter abbreviations, because we'll be working with them as we move along the course, and we're going to have to know those one-letter and three-letter abbreviations. Okay, so moving on. For the next amino acid, we have lysine. So we have the carboxylate group here. We have our amine group here and alpha carbon here. So our R group is attached down here. For the next one, I'm going to speed up a little bit. We've got aspartic acid. Now these on the left that I talked about, they were basic. So they picked up a hydrogen to become positively charged. But those are our basic amino acids. Over here on the right, we have, so this gets a little bit confusing, okay? Over here on the right, we have amino acids that had our carboxylic acid group on the side chain. So I'm talking about the side chain when I'm saying basic or acidic. Okay, I'm not talking about the amine and carboxylate that were in that those alpha carbon amine and alpha carbon carboxylate groups, because those are going to be tied up in peptide bonds. They are not going to be reactive when we form proteins with these unless they're on the ends. Okay, so for the next ones, our carboxylate group is here, amine group is here, alpha carbon is here. So this is our R group. This is aspartic acid. It's called aspartic acid because it had a hydrogen on it, and that hydrogen, when it's removed, then the side groups become negatively charged. Okay, so for the next one, we have glutamic acid. It was an acid. It had a hydrogen on it right here, has a carboxylic acid group, but under biological conditions, that hydrogen will leave. And remember, this is our R group here. Okay, that hydrogen will be pulled off and it will form a negatively charged species. So it was acidic, but now it's negatively charged because it lost its hydrogen. Okay, so moving on. So those are our charged amino acids. Moving on, we can have polar uncharged side chains, and that's due to the pKa of some of these polar groups. Okay, so like for example, this OH group has a pKa of around 16 for an alcohol, and therefore it isn't going to be lose a hydrogen under biological conditions. So these are polar because that alcohol group is going to, in this case, lend itself to be for hydrogen bonding, but it isn't going to be charged. Okay, and so here we also have an OH group on the side chain. Remember, here's our alpha carbon on all of these. And again, we have a polar group here. This one's not going to be charged because this is an amide. This is asparagine. So here we had serine. SER is the three-letter abbreviation. S is the one letter. Threonine, THR, and T. Okay, now we have asparagine, which is ASN and N for the one letter. And this has an amide bond. And then we have glutamine, which is GLN or Q, because we ran out of letters. And here's our alpha carbon, here's our R group. So again, we have an amide bond, but we have an extra CH2 group in here, making it a different amino acid. So very similar to asparagine, but a little bit longer on the R chain. All right, and so here we have some, these are also polar amino acids and uncharged, but they are some special cases. So here we have cysteine. This has an SH group. A little bit later on, we're going to talk about cysteine. It's very important because it can form what's called disulfide bonds, and those can be used to hold the three-dimensional structure of proteins. Next, we have selenocysteine, which is a rarer amino acid, but it is found in some biological systems where it has a selenium instead of a thiol group. Okay. Next, we have glycine. This one has two hydrogens here, making it achiral. And again, this is our alpha carbon here, so it doesn't have an R group other than just a hydrogen, making it achiral. It's also special because it's considered to be both nonpolar and polar, because that hydrogen is so small it doesn't really affect the polarity of the molecule. So normally we think of a carbon-hydrogen bond as being nonpolar, and that's what we have here in that alpha carbon. But since it's so small, it can also fit in with polar amino acids quite well. Okay, so when we call these polar, nonpolar, or charged, we're talking, or acidic or basic, we're talking about the R group, the alpha amine and alpha carboxylic acid groups. We don't consider those when we're saying charged, uncharged, whatever, because they are not going to be part of the backbone of the protein, and we'll see that a little bit later on. The next we have proline, and it's a special case because notice that the amine group here, the alpha amine group here, here's our alpha carbon, is tied up in this ring, in this cyclic structure, and that gives the amino acids that contain a proline, it's going to form a permanent covalent kink in that molecule, and we'll see that later on. That's important for the 3D structure of proteins and holding that structure together. All right, go down here. We'll talk about the hydrophobic amino acids. Here's our alpha carbon, and this one just has a methyl on it. Okay. Notice this chart also has the pKa, so you can look this chart up on Google. Here we have an alpha carbon, and this has a isopropyl group. Okay, and so here we have alanine, valine. Here's their 3-letter and 1-letter abbreviations for all of these. Here we have isoleucine. Here's our alpha carbon. So these are non-polar. These are hydrophobic side chains. So what's going to happen with them? They're going to orient themselves either into non-polar environments, such as in the center of a lipid bilayer where they are oriented with the tails of the lipids, or the protein will turn in on itself in an aqueous environment so that these hydrophobic groups are actually oriented toward each other. The next one is leucine. Here's our R group. Okay, here's our alpha carbon and R group in 3-letter and 1-letter code. Methionine, alpha carbon, R group. Now this one is considered non-polar because the sulfur doesn't have hydrogen bonding, so it is somewhat polar because we do have that sulfur atom there, but it's less polar than these groups listed above, and so this is considered to be a non-polar amino acid because that methyl group is going to cap it off with a non-polar group. Okay, so next we have phenylalanine. So that's our this is our alpha carbon here. So our R group is the CH3 and phenyl. Here's our alpha carbon on tyrosine, and so here we have the CH2, the phenyl, and an OH group. Now that OH group is polar on tyrosine, and so it is going to lend a somewhat different property to that molecule, but it's listed as hydrophobic, as a hydrophobic amino acid because it has this large hydrophobic region. Okay, and next we have tryptophan. Okay, and here's our alpha carbon, here's our R group. This is a large R group. Notice that these three over here on the right, phenylalanine, tyrosine, and tryptophan, are all aromatic compounds, and that's going to be important for a lot of different purposes. We're going to see some pi stacking interactions with those in the proteins. We're going to observe that they may absorb in the UV. So we'll talk about those unique properties as we get to them. Okay, so that was our overview of the amino acids. So I'll just quickly go through these slides. So here again are nonpolar. We've got leucine. Proline is nonpolar, but it does kink, and that's the one we talked about. That's a special case. Alanine and valine. Okay, so remember these 3-letter and 1-letter codes for those as well. So you can look up that chart if you wanted to. It also has the pKa of the amino acids. Here we have methionine, tryptophan, phenylalanine, and isoleucine. So here we're seeing the ball and stick model on the left and the space filling model on the right. Okay. Alright, and next we have glycine, and all we're seeing is the side chain ball and stick and space filling. Okay. Serine. Okay, so we have the these are the polar and uncharged amino acids. Asparagine, which has the amide group. Okay, glutamine. Okay, talked about these already. Threonine. These are the these are actually polar and uncharged as well, because this one has an OH on it. Threonine, cysteine, and tyrosine. So notice that the previous chart that we looked at had tyrosine listed as a hydrophobic amino acid, but this chart has it listed as a polar uncharged amino acid. So you could see there's some varying ways to list some of these, and like I mentioned, this OH group is polar, and even the aromatic ring itself has a high region of negative charge and is going to lend some polarity to that molecule. So it's kind of debatable depending on the situation what you might call this. Okay. Alright, and here are the acidic ones. We have aspartic acid and glutamic acid. Here are the side chains in yellow for those. And this slide is a little confusing here because it lists aspartic acid and glutamic acid with that hydrogen on it. So remember, these are carboxylic acids. So that COOH group is really this. Right? That's the way a carboxylic acid really looks, but your book is abbreviating it as COOH, and it loses this hydrogen here to become COO minus, right? So under biological conditions, we're going to see a negatively charged species there, and so that's the confusing thing about these slides. They list the carboxylic acids with the hydrogens, but then they list the amine group, the bases, as being positively charged, so we'll look at that in a minute. So here we have glutamic acid that's going to generally be negatively charged. Both of these will be negatively charged generally under biological conditions. Alright, next we have the basic amino acids, which have an NH2 on them. Okay, here, let me get a different color here. Okay, okay, so here and here, they do have an NH2. Well, this one is this one. It doesn't matter. These are equivalent. They have resonance, okay? But shown here, they've picked up, so this is an NH3 plus because it's picked up a hydrogen already, because it's a base, and generally under biological conditions, these are going to have a positive charge. So lysine on the left will have a hydrogen on the amine group, and the arginine on the right is usually one of these amine groups will be positively charged. But that positive charge the hydrogen can kind of, it's going to be hopping around. It's going to be picked up by other things, put back over here. So these NH2s are actually equivalent on arginine. Okay, so histidine next. We have this one of these NHs can pick up a hydrogen. One of these nitrogens can pick up a hydrogen and become positively charged. Okay. All right. And we already looked at this chart. Okay, so we're going to stop here, and we will continue this chapter in the next video.